Well, all right. The number of arrows keeps going down, so we only have 4 more times to meet, including today. So yeah, today I'm going to finish what I started last week on the last bit about sex chromosomes, and it's going to be the phenomenon of dosage compensation, which is another consequence of having different numbers of chromosomes in the same individuals. Then next week is going to be a class about methods, and mostly it's going to be three-dimensional microscopy and how microscopy and some other molecular techniques are used to study chromosome structure and structure of the genome. And then the next week is going to be mostly a kind of overview again, going over the entire class. So from the very beginning till I'll just recap what I just did the week before, with a little bit extra about the connection between chromosomes and cancer. And then 2 weeks later, we'll have our test. All right. So yeah, not much left. And to start with, there's some follow-up from last week. So remember I was talking about meiotic drive and sex determination, and I just kind of broadly said, oh, there's this link between meiotic drive and sex determination systems, and that's why sex determination can change so rapidly over evolutionary time. So you can have flies and worms and mammals, all with different sorts of, and butterflies and birds, etc., with different sorts of sex, chromosomal sex determination systems. And during that class, I said, okay, one of these reasons for why sex determination and meiotic drive are related is because the X chromosome and Y chromosomes are unequal in size usually. So if there's any competition about which one is going to get into the oocyte or into the polar body, then they can use the size of the chromosome as an asymmetry. There's essentially more chromatin on one side than the other side, and if there's any way to exploit that asymmetry, then some genes may find a way to do that. And that's part of it. But also, there's a more basic reason, which is that just regular genes that distort the segregation ratios of chromosomes, they normally don't matter so much. So you can have a gene that promotes the segregation of chromosome 4 into the oocyte. So it promotes the segregation of the chromosome 4 that it's on into the oocyte. So let's say this is chromosome 4, and let's say you have 2 versions of chromosome 4, one with this meiotic drive gene and one without. And this meiotic drive gene may say, all right, has a special effect of putting this 4' chromosome into the oocyte, whereas the just plain 4 chromosome normally goes into the polar body. And this sort of meiotic drive system is pretty common, and it can evolve in many ways. One of the most common ways is for this gene to make a sort of poison, and then there's another gene very close by that has the antidote for this poison. So when in the oocyte, both chromosomes are exposed to the poison, but only the one that has both of these genes will have the antidote as well. So in other words, if the regular chromosome 4 ends up in the oocyte, then all this poison created by its opposite chromosome will end up killing this egg, and that will lead to a large number of eggs with chromosome 4' and chromosome 4 in the polar body. So this is actually one of the most common ways that meiotic drive happens, and it doesn't actually exploit the asymmetry of meiosis. Rather, it actually this chromosome will kill gametes that contain the other chromosome. And genes like this are called sister killer genes. It's kind of a pretty metal name for a gene. One gamete will kill its sister gametes by having this poison that the sister gamete does not have an antidote to. And these things are actually rather common, and we don't really ever know about it because the poison and antidote are very tightly linked, and we may be carrying many of these genes ourselves. Definitely C. elegans, this very common experimental organism that I work with, people are finding new examples of this sort of system all over the place. There's probably dozens of these linked meiotic drive genes in there. Okay, well, what does this have to do with sex chromosomes? Well, these things are actually okay to have. So it doesn't really matter too much from the organism's perspective whether you get this 4' or chromosome 4. If something bad comes in and decides to attach itself to this driving gene, you can get some recombination that will get rid of it. So actually, it doesn't matter so much which version of chromosome 4 you get, because all the genes really are in common. So in the long run, the population as a whole doesn't really change whether they get this chromosome or this chromosome. But if this same sort of driving gene were to crop up on the X or the Y chromosome, then there's actually a big problem there, because if it's on the X chromosome, well, there's no Y chromosome in the polar body, so this isn't quite. It has to be It has to be the other X chromosome, right? If this creeps up on the X chromosome, not the Y. If this happens on the X chromosome, then which X chromosome you get may actually be a big influence over the sex ratio in the species. So if there's another, let's say there's another linked gene here that has something to do with sex determination, then depending on which X chromosome you get, then the sex ratio of the whole population may change. So you might get some sort of, anyway, if you can somehow change the segregation of the X or the X and the Y, then you can get this change in sex in the whole population, and then you would have mechanisms that correct it. So anyway, if you get something like this on the X chromosome, I think there will be strong selection to move the sex determination system to another chromosome at some point, because if one of these chromosomes is too selfish, then that can kind of hijack the sex determination system of that species. Anyway, so this normal kind of this normal kind of meiotic drive, not necessarily exploiting the size of the chromosomes, but just having this sort of sister killer system. If this gets onto the sex chromosomes, then that may lead to rapid selection of the sex determination system. Okay, I think that's hopefully that. Shed a little more light on that. And then the other question we got from last week was Anna's question about why. I said that if you are XXY as a fly, if a otherwise female fly has a Y chromosome, then that will kill the fly. But that was just that was not correct actually. Nothing I said really made that make sense, because the Y chromosome only has a few genes that are required for sperm production in flies. There are really not many genes on the Y, and it has nothing to do with sex determination, because it's not the presence of a Y chromosome that makes a fly into a male, but rather it's the ratio of sex chromosomes to autosomes, just like in C. elegans. So actually, XXYs are just normal female flies, and there's really nothing wrong with them at all. You wouldn't even notice that they have a Y chromosome. So this is very different from humans, because an XXY human is a male, of course. So there's this really strong sex determining gene on the mammalian Y chromosome, this SRY gene. But flies don't do that. They use this chromosome counting system. So actually, I was wrong. XXY flies are just completely normal females. However, if they're XXX, so 3 X chromosomes, then those flies do actually die. And the reason they die is due to the way flies change the expression levels of genes on the X chromosome. And I'll be talking about that later. But the way, okay, so I'll briefly talk about the way Drosophila, I just looked this up. So C. elegans use this pretty amazing system of counting the number of X chromosomes and counting the number of sets of autosomes and using that ratio to determine the sex of the individual. And Drosophila uses another quite similar system. And I mentioned last time that the person who figured out this system as well as this person who figured out the worm system were both in my university. So I really need to remember that I've studied this a lot and I had to kind of review it. But so they use this, Drosophila does a very similar thing. And they use this gene that's on the X chromosome. This gene is called sex lethal. Drosophila genes actually have really funny names sometimes. They have long names and some of them are quite funny or descriptive. So this gene is called sex lethal. And it's called that because if you mutate this gene in males, they will die. So a mutant for this gene, you'll see it because all the progeny will be female. So what does this gene do in a normal situation? Normally, this sex-lethal gene, when levels of this gene are high, it simply turns on the female developmental program. So it's a transcription factor, and it starts the expression of genes. And if you turn on these genes, then you will get a female. Okay, so that sounds pretty good. So this is what happens in females. But what happens in males to change this? Well, in addition to sex-lethal gene, there's another gene on autosomes. And this gene is called deadpan. I don't actually know why they named it that. But this deadpan gene, its job is to turn off the sex-lethal gene. So it says it's called a repressor. And of course, females have the deadpan gene as well, but they have 2 copies of the sex-lethal gene. So in males, there's 2 autosomes. So whatever autosome, chromosome 2 or 3, that this deadpan gene is on, that's enough to turn off this sex-lethal gene. So now there's low expression of sex-lethal, and that's not enough to make the female program, so it becomes male. But this deadpan gene also exists in females, of course. So why isn't this shutting off all the sex lethal in females? Well, there's actually another gene on the X that completes this pathway. So the X chromosome, in addition to sex lethal itself, also contains another gene called sisterless. And so when there's 2 copies of sisterless, that will turn off this deadpan gene. So now sex lethal is not turned off anymore. So this is kind of a typical genetic pathway, and pathways often have this symbol here, which means this means it turns it off, right? A positive arrow, an actual arrow, means that it promotes something or it leads to something, whereas this arrow, I don't know the name for this, but it's when people are writing genetic pathways, if you see this symbol, it means that this thing reduces this thing or represses this thing. So this gene called sisterless reduces or gets rid of this deadpan gene. And as a result, then sex lethal is no longer inhibited. So essentially, if you see 2 of these symbols, it's the same as just having 1 arrow here. It's just, it's like a double negative leads to a positive. And in males, just like with there being not enough sex lethal to resist deadpan, there's also not enough sisterless to repress deadpan. So this is why if you have 2 autosomes, 2 chromosome 2, but 1 chromosome X, then this gene product, this protein, will exist in the highest amount possible, and that's enough to turn off this sex lethal gene. So, so that's, and you'll note the Y chromosome doesn't appear in this logic at all. It all has to do with the number of copies of autosomes and the number of copies of the X chromosome. Okay, so that's about it from the follow-up. So that's how flies determine sex. And so from now on, we're going to talk about this last aspect of sex chromosome biology and what it requires at the cellular level. So once the lineage decides to use sex chromosomes, or to use chromosomes to determine sex, then this sequence of events invariably follows. That's always followed in every lineage, right? So once chromosomal sex determination occurs, then invariably the heterogametic chromosome, so the chromosome in the heterogametic sex, will start to change. And what happens eventually is it starts to degenerate. So the Y chromosome, since it's not recombining with the X chromosome anymore, in males, for instance, that all that region in between the tips of the Y chromosome, it has no way to lose bad things or no way to gain good things anymore. It just degenerates and mutates. And eventually it shrinks in size and kind of goes away. As a result, you get this condition called heterokaryotypy, which just means different numbers of chromosomes. So if you have two X chromosomes, you will have the same number of genes in 2 copies each. If you have an X and a Y chromosome, though, you will have some genes in common, those genes on the pseudoautosomal region, but the rest of the genes, you will only have 1 copy of it. And so that's actually not good. As we've seen, having different numbers of gene copies can do very interesting things, such as change the sex of the organism during development. But they can also cause a lot of just problems. If you have too much or too little of a gene, you can lead to lethality. Because genes, the amount of protein in a cell is usually really finely tuned. We need to have just the right amount in order to do the function properly. So to deal with this problem, evolution has come up with this mechanism called dosage compensation, which means that even though there's 2 copies of every gene here and 1 copy of every gene here, somehow when you look at the transcript level, when you look at the RNA coming off these chromosomes, it's equalized. So you get the same amount of RNA from in the female cell as you do in the male cell somehow. So this is called dosage compensation, because even though the gene dosage is different, this dosage is compensated for at the transcriptional or translational or some other level. So every animal that uses chromosomes to determine sex has to find some way of doing dosage compensation as well. And just as there are many ways of using chromosomes to determine sex, there are also many different ways, very rapidly evolving different ways to do dosage compensation in different animals. And I'm going to discuss 3 of them today a little bit. So probably the most complicated one is the one that we use, the one that probably all mammals use, and that is that the dosage compensation is affected by essentially taking one of these X chromosomes in the female and just turning it off. In other words, so this may represent like the number of the transcript dosage on a single X chromosome. What happens is one of these chromosomes is turned off, so this is like the off X chromosome, so that in the end, both males and females have the same gene dosage. And so this phenomenon is called XX mosaicism. And it happens during development, not at the very beginning. It happens when the embryo is about 500 or to 1,000 cells. At some point, one of the 2 X's in each cell is randomly chosen to turn off. And that one of the phenomena that results from that, as you may know, is the coat color in many animals, including these calico cats. They have some brown pigment on the X chromosome, and if one patch of skin had this brown pigment and it got turned off, then that patch of skin becomes one color. And if the other chromosome is active, then it becomes the other color. And I don't know where the white comes from. But anyway, they have different color in their fur because in those patches of fur, one of the two X chromosomes is active or inactive. So how are these X chromosomes turned off? It's actually using systems that we've already talked about. So essentially turns the active U chromatin of the X chromosome into inactive heterochromatin. Before X inactivation, both X chromosomes look pretty similar. But then after X inactivation, one of the two X's starts to really condense, and it becomes really small and dense. So if you do DAPI staining like this, you can see it kind of stands out from the rest of the chromosome. And it's even better if you use this other kind of stain you use to do chromosome banding. And you can see that these XX cells from a female, they do have this, each one has this bar body. And interestingly, so it's called, say, this inactive X chromosome has a name, it's called the bar body. And it was discovered by a cytologist named Barr, obviously, who didn't know what they were at first, but they knew that they were just in female cells. And it turned out later that this is the inactive X chromosome, because males don't have them. So, just going back, do you remember what these things are, these dark things? They're not bar bodies. So you'll see that both males and females have these kinds of spots. But this bar body is different in some way. So, does anybody remember what this is? So you don't see it in this because this stain only stains DNA. But this dark stain here is a general nucleic acid stain. It stains all nucleic acids, including RNA. And so these bodies here are the nucleoli. They're where ribosomes are made. So the nucleolus, I think it's called. Yeah. So that's where a lot of RNA is, and it's very dense. So it took a trained eye to be able to look at these cells and say that these cells all have something else going on in them that these guys don't have. But what's consistent about it is that it's about the same size in every cell, and it's always at the nuclear envelope as well. So just like most heterochromatin, the bar body, this inactive X chromosome, is also localized against the nuclear envelope. Okay, so this seems easy, right? So at some point during development, one of the two X's is chosen and turned off. It becomes heterochromatic. But that's actually a pretty complicated thing to have to do, right? Because you're essentially taking half the genes on the X chromosome, half the gene dose of the X chromosome in the entire cell, and saying you're going to shut it off forever, and it will not. This cell will not express those genes from that chromosome anymore. So one potential problem is what happens if you accidentally shut off both chromosomes? What does that ever happen, and how does that not happen? So the mechanisms behind this are still kind of being worked out. It's not exactly clear how a cell with two chromosomes is able to pick one and do something with it and not do that with the other one. But what's known is that at some point these two chromosomes have to come together to make that decision. They can't do that remotely. So these chromosomes cannot sit on opposite sides of the nucleus and communicate long range and say, okay, are you going to be the one to turn off? Because I don't want to turn off. All right, you turn off. All right, that's fine. They can't do that remotely. They have to actually come into close proximity and contact each other. Right, so, and there's this counting process, as we'll see later, and what that means is instead of saying that one chromosome is chosen to be turned off and one chromosome stays on, it's more accurate to say all but one X chromosomes in the cell are turned off. And the reason that's more accurate is because sometimes you have more than two X chromosomes. There are cases where you have 3 or 4 X chromosomes in one cell. And in those cells, all but one X chromosomes are turned off. So rather, X chromosomes by default are going to inactivate, but one of them is preserved as an active chromosome. So this choice is thought to involve some sort of feedback loop where each chromosome is expressing a certain kind of RNA, and it starts off, each chromosome is expressing about the same amount of this RNA because there's no pre-existing bias between these chromosomes as to which one is going to turn off. But it's not exactly equal. There's a slight fluctuation, and once one of them has a slightly higher amount than the other one, somehow that leads to inhibition of this one. So this one will get even less, and then this one gets even more, and this one gets even less. So having slightly more at the beginning leads in this feedback loop to one chromosome having all the transcript and one chromosome having none. And this transcript is a special RNA. It doesn't actually make a gene, it doesn't actually make a protein. But what it does is this RNA actually just sits on the X chromosome and covers the entire X chromosome and eventually leads to the inactivation of this X chromosome. But one chromosome doing this will lead to the other chromosome not doing it. So the mechanism of inactivation is linked to this feedback loop such that all the inactive chromosomes get covered with this particular kind of RNA, but one chromosome is left open and does not get coded with, well, it might get coded with a different RNA, but at least it doesn't get coded with this repressive RNA, so it can stay active. This counting mechanism, the fact that it's actually one chromosome is left not inactivated, comes from the kind of sex chromosome aneuploidy data that talked about before. So remember, humans can tolerate different numbers of X and or Y chromosomes in their cells to different degrees. Can tolerate them much better than autosomes. And now this particular method of sex chromosome, of dosage compensation, can actually explain why we can tolerate different numbers of X chromosomes. Because the two typical cell types, XX and XY, are very simple. So the XX cells have one inactive X chromosome, one bar body per cell. XY male cells, we do not have any bar bodies. However, there are other options. So XO individuals, so people with just one X chromosome and no Y chromosome. Of course, there's no. It's just kind of like the male situation. There's no counting. There's no recognition of multiple X chromosomes, and they also have no bar bodies. XY individuals, males with 2 X chromosomes instead of just one, they do this X chromosome inactivation. So it's not that X chromosome inactivation is not incompatible with having a Y chromosome. The Y chromosome does not participate in this. And then really interestingly, triple X females with 3 X chromosomes actually have 2 bar bodies in each cell. So if they started with 3 of these chromosomes, then somehow 2 of these chromosomes end up covered with this RNA, and one of them is not. So that's the right thing, right? That's the way you would want it to go. If you have if you want to have just one X chromosome worth of genes being active, then every additional X chromosome you want to inactivate. So that's a pretty interesting situation there. So somehow this mechanism is general enough that it can turn off every X chromosome except leaving one. And this is why aneuploidies for the X can be tolerated more. So most aneuploidies, having 3 of any of the autosomes, except for a very small number, including chromosome 21, will lead to lethality, immediate lethality in embryonic development because of this gene dosage effect. Even have 3 copies of genes where you were normally expecting to have 2 is enough to kill the developing embryo. That's how sensitive gene dosage is, and that's why dosage compensation is required. But for the X chromosome, it takes quite a lot of, it takes having 4 or more X chromosomes before you really start to see some kind of phenotypic effects. However, that's not exactly true, right? So there are some differences, right? XO females have what's known as Turner syndrome. They have some health problems, and XXY males also have some health problems. They're not completely normal-looking, healthy people. They do have some health problems in addition to the infertility from having the incorrect number of chromosomes. So if these chromosomes are turned off, then why do these syndromes exist? It seems like it's the perfect solution. If you have too many X chromosomes, you just turn them off, and then there should be no problem. So why are there problems? Does anyone have any idea? Why might there be some health problems? Let's take just let's take the XO compared to XY. They have the same number of genes on the X chromosome. So why might XO females have a problem? Or compare, maybe this is easier. So typical male with the XXY Klinefelter syndrome male, they have the same number of active X chromosomes, so why is there some problem?
So it's not switched off till later
Yeah, actually, that may be that may be one reason. That's not the reason I am most familiar with. I think those first 500 divisions, not 500 divisions, but the first 10 or so divisions, I don't think there's a difference at that stage. I think that is not detectable. I don't think there's any problem at that point. It actually has to do with later developmental processes. But you're right. That is a possibility because until one is turned off, those first 10 or so divisions, they do have a different dosage. So that's another thing I should look into. But that's not the reason it's commonly cited. But that is a good guess. And there's probably some truth to it. But so the answer you're more likely to hear is that although we say that the X is inactivated, it's actually not 100% inactivated. There are many genes that escape inactivation. One of them being this, of course, this gene that produces RNA that covers this entire X chromosome. The chromosome needs to keep producing that RNA over the course of its entire life, of course. So obviously this bar body is still active because it's still producing this RNA that represses itself. But there are other genes that are also not inactivated on the X chromosome, which means that these bar bodies that I showed you, even these really condensed chromosomes, they're still actively transcribing some of their genes. Probably they're not heterochromatic. So even these heterochromatic X chromosomes still have some euchromatic parts. And can anybody guess what some of those genes might be or where they might be?
RNA creating region.
So you mean in the same region as this RNA? Yeah. I think that's true. I think there are some other genes right around. So this RNA is called X-cyst. This gene is called X-cyst that makes this RNA. And probably right around this region. You're right. It's probably euchromatic for some region around it. I think that's probably some of the genes might be around there, yeah. But there's other places too where you might expect genes not to be inactivated. Yeah.
Power region?
Yeah, that's the other place, and that's the other kind of wrinkle in this dosage compensation story, because as we know, there are genes that are present on the Y chromosome as well as the X that are identically expressed between the Y and the X, and they're inherited, so they do not present any sex-linked inheritance, which means they're not turned off on the Y, they're not turned off on the X, but they're expressed biallelically like any other autosomal gene. And so any gene that's in here, we expect to always have two copies, whether you're a male or a female. So XY cells will have two copies, XX cells will have two copies on the X. So that's a cell is normally, and there's only about 24 genes. It is a very small number, but some genes are known to reside in those regions and be required for normal health. So for instance, this XXY Klinefelter male actually has 3 pseudoautosomal regions. So it's getting 3 times the gene dosage, and the same for this XX female. There's also 3 pseudoautosomal regions. Even though I don't think these females have any problems at all, actually. But where you do get health problems if you don't have enough of that gene product. So not having enough of those genes in the PAR regions is also bad. So yeah, instead of having 2, this gene dosage is only 1, and these gene dosages are 3. So those are the cases that lead to health problems, is because this X inactivation is not complete. So in that sense, mammals have a pretty baroque, complicated dosage compensation system that still can lead to some developmental problems if you have the wrong number of X chromosomes. So I'll briefly talk about what flies do. So even though flies have X's and Y's just like us, their Y chromosomes are even more useless compared to human Y chromosomes. They literally have nothing on the Y that is not needed, that's needed by females, and they only have a few genes involved in sperm production. So XXY females that I mentioned, they're perfectly healthy, not dead. They're fine, and you can make an XX male, which you do by having XX and then just the one gene, where you just you mess with the sex determination system so that you make an XX male. They're also healthy, except that they're sterile because they can't make sperm. So that's the only thing they need the Y chromosome for. Other than that, they're completely healthy. But they change their expression on the X chromosomes in a completely different way than humans. So instead of randomly choosing an X chromosome to inactivate, what they do is they notice that they find themselves in a male body. So if the X chromosome finds itself without a partner, then these other proteins called MSL proteins, they bind to the X chromosome, and what they do is they increase the level of transcription. So they take this euchromatic chromosome and make it even more euchromatic in a way. They just increase the transcription level by a factor of 2, making the total amount of RNA coming from the single male X roughly equivalent to the total amount of RNA from the 2 female X's. So that's how flies do it. And then the last thing I'll talk about today is how worms do it. And there it's, you might think they could use the fly system, but they actually do the exact opposite of what the flies do. So instead of taking single X in a male and turning it up by 2, they take the 2 X chromosomes of a hermaphrodite and dial them down by a factor of 2. So they don't completely shut off transcription of one X, rather they take both of them and do the exact same thing, which is just reduce transcription by a factor of 2. Yeah, so how does that happen? Well, let's go back a bit and remember a little bit about the sex determination system of C. elegans. So there's this gene called ZOL1 that exo animals need, and it's the master switch that turns on male development. And these red bars are these things called the X response elements. And the more of those you have, the more you are turning down the expression of ZOL1 and leading to hermaphrodite development. And remember, two X chromosomes have many of these XREs, many of these repressive X response elements. So they really turn down ZOL1. And then if you have a fragment of the X, what matters is how many of these XREs you have, and that determines the power of hermaphrodite generation. But this ZOL1 gene itself is interesting. So it's called, remember, ZOL1 stands for exolethal. So if you mutate this ZOL1 gene in males, then they die. So the question is, why do these males die? They start male development. They can do this counting of X and autosomes, but they don't have, they don't live very long. And so why do they die? And it turns out that the reason they die is because, well, I'm kind of getting the story backwards. What people originally wanted to do is say, okay, we know that these ZOL1 males die. Let's mutate more genes. Let's cause further mutations in addition to ZOL1. Let's try to mutate more genes and get a situation where ZOL1 males can live. And so why might you do that? Well, what's happening is, so death is the end result here, and maleness is the beginning. So normally males live, of course. Males have, males use ZOL1, and then they are fine, they're happy. Let's say this leads to life, okay? If you don't have ZOL1, then you don't have this. But it doesn't just go directly to death. There's probably some other gene X and some other genes that eventually initiate whatever process is killing these males. So the idea is, if you cause further mutations, you can delete these other genes that cause ZOL1 males to die, and instead you will go back to living males. So you'll have males without ZOL1, but without these other genes, maybe they can live. So this sort of experiment is called a suppressor screen. So a screen is just an experiment where you make a lot of mutations and look at everything you get and try to figure out something about these genes by looking at different mutations. And specifically, suppressor is when you already have one mutation that leads to death or some other problem, and you suppress this problem. So you're suppressing the death of worms by mutating some other gene. And in fact, this is a really nice way to do such a screen, because since normally you're leading to death, so on this normal plate with worms on it, you will not see any living male worms. So all you have to do is mutate genes and look for living males on the plate. And if you find them, then you know that you actually succeeded in knocking out one of these genes. So okay, so this is, and when you do this, when you suppress that, it's called, the term is that you would rescue the lethality. So if there's a bad phenotype like death, and you say that you have rescued this phenotype by mutating this gene. And so this experiment was done a while ago, and many genes were found actually that suppressed this lethality. And what these genes turned out to be related to the dosage compensation pathway, which means that the reason ZOL1 males would die in the first place was that they were not able to turn on, that they turned on this dosage compensation pathway spuriously. So, right. Yeah, so ZOL1 is normally turning off this dosage compensation pathway in males. But then, so when you mutate ZOL1 in males, you are allowing dosage compensation to act, right? So this is what normally happens. Normally, there's equal amounts of RNA coming off the single male X and two hermaphrodite X's, because this hermaphrodite X is turned down by 50%. This dosage compensation does not happen in males because ZOL1 is preventing it from happening. But if you lose ZOL1, then even though the body wants to develop as a male, the chromosomes pretty much think that they are in a hermaphrodite cell, and they do also turn down by a factor of 2 the amount of transcription. So they don't have enough gene product, they don't have enough proteins that are coded on that X chromosome. And that is what leads ZOL1 males to die. So what are these gene X's that were found? So very interestingly, let's see. Yeah. Well, so I'll just show you show you a diagram of some of these genes. Like all C. elegans genes, there are 3 letters and a hyphen and a number, and so they're really kind of indecipherable. But I'll tell you about what some of these stand for. Okay. So anyway, these are the genes that were found as a result of this successful screen. So many different genes were found, and they all, or most of them, ended up making proteins that were part of this same complex. Often in cell biology, you get many proteins working together to do one common function, kind of like the 4 histone proteins all come together to make this histone octamer. Similarly, all these different proteins come together to make one complex called the dosage compensation complex. And what's really interesting about this is that we've seen, at least this part, the dosage compensation complex, is very similar to another complex that we've seen before, which is the condensing complex. So remember that when chromosomes condense, part of how they condense is that this condensing complex makes these rings around chromosomes that cause these loops to come out. So these rings are actually made of these proteins that have this sort of shape. There's a base, and then there's 2 arms that can link together. So it's not really a ring, it's really something that looks more like this. So there's a base, and then these 2 arms, and the DNA goes through this loop made by that protein. Well, the genes that could suppress the lethality of ZOL1 also made this sort of structure. Further experiments showed that this is exactly the role they have on the X chromosome, is that they condense it a little bit. So they just sort of turn the X chromosome a little bit more heterochromatic, but not quite as heterochromatic as the bar body. So it's still fully transcriptionally active. Every gene on the X chromosome is still able to be transcribed, but just by kind of making it a little bit more dense, it makes transcription just twice as difficult, and so the number of transcripts is also reduced by a factor of 2. So what do these things stand for? Well, some of them were new genes that no one had ever found before. And so these were called SDC something. SDC stands for sex determination and dosage compensation, because they're actually involved in both. Sex determination. Is SD, and then dosage compensation, fortuitously also started with a D. So those are the SDC proteins. But the interesting case are these dumpy. So DPY actually stands for dumpy, which kind of means short. And there are many dumpy mutations that were already known. You can see this one goes up to 28. I think there's at least 50 different dumpy mutations that turn a normal, long worm like this into kind of a short worm with the same width. So this is what a dumpy worm looks like. And why it's interesting that some of these dumpy genes were found as suppressors is that if you have if you cross if you have a worm with two X chromosomes, but for whatever reason they can't pair or recombine their X chromosomes, then you get a situation where you can have, in the gametes, you can have either no X chromosomes, or a single X chromosome, or two X chromosomes. And that happens in the sperm as well as the eggs. So the combinations you can get, so this is, let's say that there's a mutation where the X chromosomes cannot pair and recombine. You get here XO, this will be a male. XX is a hermaphrodite. If you have two X's and O, this is also a hermaphrodite. So this can be filled out pretty easily. Of course, if there's no X chromosomes, then this is dead. If you have four X chromosomes, that's also dead. But interestingly, every example where you have three X chromosomes in a C. elegans, those are hermaphrodites. If you have three X chromosomes in C. elegans, they are hermaphrodites, but they're also dumpy. So, in other words, having the incorrect dose or having too much X chromosome transcript coming off was known for a very long time because people did these sorts of crosses a very long time ago, and these worms were always dumpy. So something about having too much of X chromosome transcript during development led to this sort of body shape. And so that's why, in addition, so three of these genes, four of the genes that are involved in dosage compensation are what's responsible for this dumpy phenotype. So that explains why when you have three X's, those worms become dumpy, because you get the same sorts of defects that you get when you miss one of these dosage compensation proteins. So that's kind of how dosage compensation works in these three different systems. And I think more importantly, it's really a nice illustration of how you can just start with something very simple, like a single gene that leads to male death, and find out something really cool about dosage compensation just from this simple genetic screen where you look for living male worms. And yeah, that will end today's class, I guess. So if there's any questions about this, please ask now or afterwards. Yeah.
With the mammalian system, is it completely random? Because like when just disorders like haemophilia, you can have female carriers who don't have the phenotype.
Right. Yeah, in that case. So you say, I'm not that familiar with this hemophilia, but is it mostly carriers? Is it 50-50 to be a carrier, or is it?
It's you can have female carriers, but they won't have
Right.
the disease unless they've got both X's faulty.
Right. Yeah, I think it is. So I see. So in that case, they only get the disease if both X's are faulty. So okay. Yeah. So in that case, it must not be random. It must be it must be in whatever the hematopoietic lineage or whatever lineage of cells is responsible for this hemophilia. Perhaps they can, perhaps that must, yeah, I don't know what gene is responsible. Yeah, it's hard to see how that mechanism could work, but yeah, it sounds like it couldn't possibly be random. Unless, okay, so I guess one way it could be is if so the choice point, if the X inactivation happens well after the tissue is established, right, then it wouldn't matter. Maybe half of the cells have the faulty copy, but if it's like a cell non-autonomous effect, then even if only half your cells have it, then you would still be compensated for it, I guess. I mean, that's kind of how color blindness is also male specific. So there's X-linked color blindness, where if you lack the pigment on one of your X's, females can see color just fine, but the males will not be able to see red or green. But of course, it's X inactivation. You would think, why can't females be colorblind too? But so their retinas are actually a mosaic of cells that have the color receptor and cells that don't have it. And even having it in half the cells, as long as they're this mosaic, it can compensate for missing the color receptor in half the cells. So probably I don't know which cells it is that is responsible for this particular hemophilia, but yeah, maybe just having it on in half the cells is enough, I would think. I think it's also in all, it's not a kind of permanent inactivation in all tissue types. I think it gets inactivated at this 500 cell stage. Then in some tissue types, you can reverse and then reestablish itself. Maybe because of this sort of thing that they don't want to lock into this decision quite so early. So some tissues it can get reversed. So we have a finer mosaic in that case. Yeah, that's a good question. Okay, none other questions then? Yeah, thanks.